Newly designed synchrotron X-ray fluorescence nanoprobes permit visualization of the elemental distribution of a sample. The use of cryogenic approaches is mandatory to slow down X-ray radiation damages. Therefore, a rapid cryofixation is essential for obtaining frozen hydrated cells to optimize the preservation of the cell structure and its chemical integrity.
The specificity of the cellular preparation for synchrotron studies lead to the optimization of this protocol for acquiring frozen hydrated cells monolayers. Visual demonstration is important for understanding how to manipulate the cells monolayer samples to achieve reproducibly cryofixed cells embedded within a thin layer of ice.
To prepare a silicon nitride membrane support, open the capsule containing the membrane support and gently squeeze the capsule to lightly loosen the support. Use thin tweezers to grasp one corner of the silicon frame, taking care not to touch the silicon nitride membrane in the center of the frame, and place the frame into an uncovered petri dish in a laminar flow cabinet. Then UV sterilize the membrane for 25 to 30 minutes. At the end of the sterilization, add 10 microliters of attachment factor, such as poly-L-lysine. Incubate for 25 minutes at 37 degrees Celsius and at 5% carbon dioxide with humidity. Then, fill the wells of a sterile 48-well plate with 200 to 250 microliters of 0.22 micrometer filtered ultrapure and ultratrace water per well. Use fine tweezers to carefully submerge the membrane vertically with 3